I came to KTH campus quite a long time ago, so I thought it was appropriate to tell you a little bit how it was in previous times here. So I was not here at 1917. I was not able to be here at that time, and I was not here when Campus 50 was celebrated either. If it was celebrated, I don't know. But at that year, I started my undergraduate studies at Stockholm University. And then 3 years later, that's why I have 47 here, it was time to select a topic for PhD studies. And I hesitated a lot between both physics and chemistry. I had studied theoretical physics and also chemistry. Very different subjects, but you might know that a very famous professor of theoretical physics, Inga Fischer-Hjalmars, that was her two topics. Anyway, I decided to go for organic chemistry. And what is then so interesting with organic chemistry? Yeah, actually today you can do very many things with organic chemistry. What is particularly important is that you can understand very much about the nature around us. You can understand a lot about life in nature and in particular about human life. We know very much about the mechanism of cellular processes, and all that is organic chemistry because we know a lot of these things at the molecular level. And you can see this from several Nobel Prizes from recent years. A few years ago, it was the mechanism and the function of the ribosome in the cell. And that means that we will know more and more about life. And we can also mimic a lot of these processes by constructing new systems with organic chemistry. What we also can do is that we can cure, we can make pharmaceuticals, because many of the pharmaceuticals we use are small organic molecules. And we can also make a lot of other compounds that we need in our daily life. What you see here is, for example, menthol, which is quite a simple molecule. But, you know, every year between 25 and 30,000 tons are prepared industrially of menthol, used as a flavor and addition to a lot of things we eat and drink. So this is quite fascinating. When less was, of course, known at that time when I wanted to start my PhD studies. So I was at Stockholm University, and one area that had developed quite a lot, or that started to develop, that is what is called organometallic chemistry. You know, carbon compounds is the basis of organic chemistry. You have carbon, hydrogen, and many compounds contain oxygen, nitrogen, and a few other elements. But what people started to use then was organometallic compounds, meaning compounds which have a bond between carbon and a metal. And that proved to be very efficient because you can activate molecules in that way to make them react more easily. And you can also determine the site of activation by attaching the metal on a certain place. And I thought that this is certainly the topic of the future. This is what I wanted to do. But the problem was that there was nobody at Stockholm University working with this type of compounds. But I knew there was a group at KTH. Actually, there were two research groups at organic chemistry working with compounds containing lithium, copper and palladium. So I thought that's what I wanted to do. I was not brave enough to just go in on the campus here, but I saw an announcement in the newspaper. That group, in one of the groups, they wanted to have a technician. So I applied for the position as technician, and I got it, and it was transformed into a PhD position quite soon. So I came exactly where I wanted to be. At that time, chemistry was quite different. So this is a terrible example of a hood. My hood did not look this way. A lot of the chemistry was done on the bench, and at that time the bench were of wood, and when you spill something, some chemicals on the bench, it was absorbed by the wood, and that gave the very characteristic organic chemistry odor to all organic chemistry labs. We don't have that anymore. Now we work in very proper, beautiful hoods. And of course, the safety was also different. We had gas burners when we wanted to heat something. We could smoke in the lab if we wanted to. But it was, anyway, although we had quite poor equipment in many cases, we developed quite well, and we had a very good atmosphere. We helped each other and worked long hours and liked really what we were doing. We had literature seminars at 8 o'clock in the evenings, etc. So it was quite a nice time. But many things were, of course, much more difficult than they are today. When we wanted to write a manuscript, we had, of course, to use a typewriter. If you wanted to change the places of 2 paragraphs, you had to cut the paper and glue it together. And if you made some mistake, you put white tape on the line, and then you were typing on that line again. Also, making slides for conferences was much more time-consuming. And the machines, the type of machines that we used, had not letters big enough, so we had to use them to make them by hand. And also searching the literature was very time-consuming. Now we have the chemists use what's called SciFinder. We press a button, we draw a structure, or we put in some keywords, and after some seconds, we have the result. Then we had to go to the library. We had meters after meter with shelves with chemical abstract service. And then we had to look up and it took at least half a day, sometimes 1 day, to go through all the volumes. The last year was 1 volume per week. Then it was a big success when the library got to computerized research. Then you could make an appointment with the librarian, and you went there, you made a research profile, and a couple of days later you could get your result. So things were quite different. So then turning back then to organic chemistry, one thing which is really fascinating with organic chemistry, that is that you create your molecules. You design molecules, and then you prepare them. Sometimes it's very difficult, but nowadays you can prepare almost any structure with the correct 3-dimensional structure. So it's a lot of architecture that is done, and this famous French chemist, he just noted that chemistry is nice because it creates the objects that it uses. So actually, organic chemistry, as I also mentioned in my first slides here, has become very important for biology. So this is quotation that the advent of organic chemistry shaped the world, which it did to a large extent. And biology is indeed dependent on organic chemistry because we need to make the molecules, we need to mimic processes in order to understand them, etc. And organic synthesis is the foundation of biotechnology, but also nanotechnology, because you can prepare a lot of new material with synthesis and with organic synthesis. So for pharmaceutical industry and for material industry, it's a very important subject. So my research has mainly been about catalysis, still using metals, but catalytic methods. So why catalysis? And nowadays, you want to make what is called green chemistry, that is environmentally benign. That means that of course, you don't want to put in too much energy, not cooling or heating too much. You want to use solvent and chemicals which are not too poisonous. And first of all, what may be particularly important, that you should not produce any waste. Because what you put in should come out as a product, and nothing else should come out in the ideal case. And therefore, catalysis is important, because with the catalyst, you can use a small amount of that catalyst in order to produce a large amount of the product you want to have. In contrast, if you have a reagent that you need to use in a large amount, then the reagent is left over after the reaction. So what I have been doing mainly in my research is to use metal complexes, and they typically contain a metal which is surrounded by an organic compound, and that organic compound then determines the properties of the catalyst. So very often we do a lot of modification. We design these ligands and we do modifications. And then, of course, we can change the metal here, because organic chemistry now can use the entire periodic system, and in particular, transition metals, which are important. So this cartoon then shows us what can happen here. So we have this then symbolizes the ligand which surrounds the metal, and we have a metal center here. So this is the catalyst. And then we have some organic structures that are going to combine in this case. So they can both bind to the metal, and transition metals are good because they have the ability to bind to many different groups at the same time, because they have many available orbitals that they can use for chemical binding. And then, while coordinated to the metal, these two form a bond between them and leave here, and this can go on in a cycle. So my work has mainly, or almost exclusively, been concerned with chirality. And chirality, that means that when an object is chiral, it means that it's not identical to its mirror image. Of course, everything has a mirror image, except vampires, which don't have mirror images, but everything else. And of course, a mirror image of an object can be identical to the object or different. Like my hands, which of course, one hand is the mirror image of the other, but they are not identical. And that's important in all biological systems in particular, because they have different properties then. Just to simplify this, if you want to have a molecule that is selective for one receptor in the body, just to make a simple picture here, so if we think that this is a molecule, then if I put this in the right glove, or if I put the other one in the right glove, is of course quite different situations here. And that is just a simplified picture of what happens in the body. So this is something very important, and it's a very, say, topic that many persons work with around the world. And very often when you make what is called asymmetric synthesis, that means that you want to make one mirror image and not the other one. Normally when you make a synthesis, you get equal amounts of the two. So you have to make certain catalysts then that favor one of them. And very often that catalyst makes small mistake. So you don't get 100% of the one you want to have, but you get maybe 80% if you have a bad catalyst, or you get 98%. And if you want to use this as a pharmaceutical, for example, it can be deadly poison. So you need to have them completely pure. And one thing we have done in recent years is that we have developed a method whereby we can correct this. So the wrong enantiomer then, or the wrong mirror image, that is the minor product, that goes back to starter material again and gets a second chance in the reaction. And then this is all corrected. So it's a cyclic process, which has proven to be very useful. So I will just show one example of a typical compound. So normally what we do is that we develop methodology to make new compounds. And then we apply this methodology to compounds which may be of interest. And often we try to apply them, for example, to pharmaceuticals. And this is a pharmaceutical called sulabegron. It's not a licensed pharmaceutical yet, but it has been a clinical trial for quite a long time. And now it's in large-scale clinical trial. It's used particularly against overactive bladder, that is incontinence. And since we live longer and longer, many of us will suffer from that in the future. So it's very important pharmaceutical that's developed. And this seems to be acted in a very good way. And the mode of action is different from that of previously known compounds. There are some analogues of this which are known. So it binds to what is called the beta-androgenic receptor, which is a transmembranic structure like this, which is what is called a G-coupled receptor. A G-coupled receptor was the topic of a Nobel Prize a few years ago, a Nobel Prize in chemistry. And what is its in a way, quite simple molecule. It's not so many atoms here. What is the crucial in this structure is this OH group here, because this means that it points towards you. If it would have pointed on the other side of the screen here, then it would have been the mirror image, and that could then maybe be detrimental. I don't know if that has been tested. So this is just one example of methodology and molecules that we have developed. So the future of chemistry, I would say that it's very, very bright, because we can do more and more. And nowadays, we can even make systems which can correct themselves, which can undergo evolution, etc. So more and more sophisticated. And often nature is the say what we learn from nature in order to mimic and get inspired of nature very often. And this is a quotation which I think is very nice from Ben Fringa, who was a Nobel laureate in chemistry from last year, the one who made the small molecular machines. And that's also another type of organic chemistry which is very fascinating, called supramolecular chemistry with small machines, which is also an area which will develop very much. But he said that for chemistry in general, the future is extremely bright, that many of the key issues related to health, food, sustainable materials and processes, just to mention a few with major impact on society and economy, arguably will not be solved without major breakthroughs in chemistry. So there is a lot of future in chemistry. So then, is there a future in KTH then? Of course, a lot has happened. And KTH has evolved to a prominent research institution. And I think we have good facilities, we have a good infrastructure, and in many ways it's good to work here. But I think it's also quite tough, in particular for basic sciences, to get enough funding and so. So it's a tough time that we have here. So we will see what will happen in the future. It's said for the future that KTH will rest on 3 pillars. But my view is that if something rests on 3 pillars, it can quite easily tip. So I think that a 4th pillar, which is very stable and thick, is needed. And I would like to call that 4th pillar excellence, because that is what we need. We need to be excellent in order to have a good future. Thank you very much.